Okay, hi grade 10s. In this short little video, I'm going to explain two structures that are only found in animal cells, and it's the vesicles, the lysosome vesicles and centrioles. And it's on page 50 of your textbook, but they've only outlined vesicles there. So you need to have a look at your table summary for centrioles. Okay, so lysosomes only found in animal cells. So you may remember that they are little vesicles that break off the Golgi body, and they contain digestive enzymes. And digestive enzymes are a type of protein that were made at the ribosomes on the rough ER. So these vesicles pinch off the Golgi body, and they can fuse with old organelles like mitochondria, and then the enzymes will digest the mitochondrial structures, the membranes, and those substances can be reused, recycled. Or an amoeba that takes in the food organism, the lysosome will fuse with the food vacuole that's created and digest the organism, and the nutrients will be absorbed. Or even a whole cell. When cells get old, the lysosomes help to break them down. So an interesting example is when we are a fetus, our fingers aren't separate, they're actually joined by a web, and then something called programmed cell death occurs, and these cells have got lysosomes of the web, and it will break down those cells so that you end up with separate fingers. So that's why you sometimes get people born that have still got some form of webbing in between their fingers. Okay, they're also abundant in white blood cells, because if you think about it, they're part of your immune system. They have to take up germs like bacteria, and they break them down. So they need lysosomes to break down those invading germs. It's a very simple structure. It's a membrane with enzymes inside. Okay, not a double membrane like over here, but the protein that at least the proteins or enzymes are found inside. All right, and there's a video on this as well, which I will upload. So here you can see the ribosomes on the rough ER, the proteins were made, little vesicles break off, join with the Golgi, break and join, and then eventually when the vesicles are mature, they break off, and now we can call them lysosomes. So here we've got a lysosome that's fusing with an old mitochondrion to break it down, all right, and then those molecules are recycled by the cell, and here we've got a lysosome that has fused with a food vacuole. So if it's an amoeba, it's taken up, it's engulfed its food organism into it, becomes a little food vacuole which moves through the cytoplasm, and then a lysosome fuses with it, and the enzymes are released and digest the organism, and the nutrients are going to be absorbed. Okay, and there's also, you can do the activity on page 51 regarding animal cells. Okay, then the last structure. So we have discussed this already. So in the cytoplasm, there are two cylindrical bodies near the nucleus at right angles to one another. They're not really well understood. People know what their structure is, or scientists know what their structure is, but they're not really well understood how they work. So they form what we call spindle fibers during cell division, mitosis, and they help to pull the chromosomes apart. I'll show you on the next slide. So that before cell division, okay, when the cell's undergoing normal processes, there would be two of these in a patch of cytoplasm, and that patch of cytoplasm is called the centrosome. So here is the centrosome, and there's the one centriole, you can see it from the side, and the other one, we can see that has been cut through transversely. So you're looking almost like looking down a straw, but they're made up of lots of little microtubules joined in a very specific way. And they're only found in animal cells. So when cells divide, we'll look at that when we do the section on mitosis. These centrioles, they actually replicate, they copy themselves, and then there would be two pairs. So there was one pair of centrioles. They replicate and they move to opposite ends of the cell, and protein fibers called spindle fibers develop between them, and they will help pull the chromosomes apart. They contract and pull the chromosomes apart when the cell divides. All right, now the last part regarding cell structures, I have mentioned this before. It's just something interesting. It's new. Using different kinds of microscopy methods, scientists are actually able to see the cell cytoskeleton. They call it cell art. Cytoplasm is not just jelly-like. There's also these fibers passing through, giving structure and shape to the cell, and the organelles can actually move along those fibers, like little tram lines. So that video that you watched about chloroplasts moving, then those chloroplasts would be moving along the cytoskeleton, and chloroplasts tend to move to the part of the cell where more sunlight is shining on them. Okay, there's some more pictures of the cytoskeleton. Okay, could be put up on a wall as a piece of art almost. All right, and that's basically it. So with regard to cell structures, the next video, the last section on cells, we are going to look at the processes of diffusion and osmosis. So I'm going to revise that because we have new pupils in our grade, and then that will be the end of cell structure. And then after that, we move on to cell division, mitosis.